All right, so in this video, we're going to be looking at the microscopic anatomy of cardiac muscle cells. And in the last video, we talked about kind of just the gross anatomy of the heart. So we talked about heart chambers and valves, the direction of blood flow, as well as the coronary vessels. Here, we'll kind of focus more so on the microscopic anatomy of the heart and talk about cardiac muscle tissue, as well as the electrical properties of cardiac muscle tissue. So what we find, you guys, is that this will be a kind of a nice review from our muscle discussion back in AMP1. So if y'all remember, we talked about like striations and sarcomere contractions and that kind of stuff. So that'll come back again here in cardiac muscle. Now, so there are some differences, though, between cardiac muscle and skeletal muscle. And for one, they're both striated, right? But the major difference here is that these muscle cells are short, whereas the the skeletal muscle cells were very long, right? Like skeletal muscle cells will go the whole length of that muscle, whereas cardiac muscle cells are very short and they're branched, which means they can kind of split out and then connect with multiple cells. So they're interconnected. They usually have about one or two nuclei at the most, and they also have an endomysium, which has lots of capillaries that can kind of supply those cells with nutrients. We also find that there's T-tubules in these cells. If y'all remember going back to AMP1, we said that T-tubules were those kind of tubes of sarcolemma that went deep into the muscle cell. That way action potentials can get deep into the muscle cell to excite that sarcoplasmic reticulum. We still find sarcoplasmic reticulum here as well too, guys, except for the SR that's in cardiac muscle isn't as extensive. It's a little smaller. And what we'll find is that some of the calcium that's involved with muscle contraction in cardiac muscle actually comes from outside of the cardiac muscle cell, whereas back in skeletal muscle, all that calcium we talked about came from within the SR within that muscle cell. So that's also different here, is that the calcium comes from extracellular fluid, not only the sarcoplasmic reticulum. Okay. We also find too, guys, that there's lots of mitochondria here. So they can occupy about 25 to 35% or so of the cell volume, which means that these cells are highly metabolically active. So if we look at this slide here, what we see then is this is cardiac muscle tissue. It's pretty distinct from skeletal muscle tissue because these cells are branched. You guys can see that, and it's what gives us sort of a disordered appearance. And then all of these are called intercalated discs. So these intercalated discs are basically the places where cardiac muscle cells meet. So for instance, like this would be one cardiac muscle cell, and then you have an intercalated disc here where it meets up with another cardiac muscle cell. You guys can see that. To me, it kind of looks like bamboo or something. Like there would be like a little joint where each of those little bamboo segments meets. But in that intercalated disc there, the reason why it's dark is there's lots of protein. And the protein you find in an intercalated disc are gap junctions and desmosomes. So this is also going back to AMP1 because we said that gap junctions were little protein tubes. You guys can see that here in this slide, where basically molecules can flow directly from one cell into the next, right, through these tubes, the gap junctions. This plays an important role in your cardiac muscle because this is how electrical currents spread through the heart, is they go through those gap junctions directly from one cell into the next. So for instance, if we were inside of a cardiac muscle cell here, right, like in this room, that door would be a gap junction, and we could just easily flow or walk through into the next room. And the next room over there could be like another cardiac muscle cell, right? But imagine if we were like the electrical current, what that means then is that we can move easily directly into the next room or the next cell. But that would be through those gap junctions, which are the little tunnels, you might say, between cells. Now, there's also lots of desmosomes here too, you guys, and these desmosomes are involved with strength and support. So if y'all remember, desmosomes hold cells together very strongly, but that also makes a lot of sense here because we want to be able to hold these muscle cells together strongly such that the muscle doesn't tear as it starts pumping blood, because it has to produce a tremendous amount of force. So remember, these intercalated discs have desmosomes and gap junctions. Gap junctions were what are these little protein tubes that allow these cells to be electrically coupled. And by being electrically coupled, what that means is that if you have an action potential in one cardiac muscle cell, it'll directly spread into the next cardiac muscle cell through those gap junctions. Now, the desmosomes hold these cells together strongly, and what this allows for, though, you guys, is the whole heart to function as like one big cell. So we said that the heart is a functional syncytium. So a syncytium just means like a together cell, you know what I mean? But the reason why it functions like a one big cell is that if you have action potentials begin in one area of the heart, they will spread throughout the whole heart, right? And the reason being is that all of these cells are linked up in some way through their gap junctions, through a very specific pathway and pattern, which we'll see here in a little bit, okay? So it functions as one coordinated unit. So if we zoom in on this cardiac muscle cell and look at its anatomy in better detail, what we see here, guys, is this is the cardiac muscle cell, right? And they're pretty short, right? So you see them from here to about there is one cell. And there's the nucleus. And if you guys zoom in on this and we look inside of this cell, what we'll find then is there's still the myofibrils, like we talked about in skeletal muscle. So we still have our thick filaments and our thin filaments here. Remember, the thick filaments have our myosin heads, and our thin filaments had things like troponin, tropomyosin, and actin. And if y'all remember, troponin was a calcium-sensitive protein, so that once calcium gets released and calcium binds with troponin, troponin pulled on tropomyosin, exposing those actin binding sites such that the myosin heads on the thick filament could reach up and grab on and begin the power stroke cycle, right? And what that does is it actually allows for all these sarcomeres to shorten. And so remember, a sarcomere is the functional unit of muscle contraction, and it's basically the region between each Z disc here. So this is a Z disc, there's a Z disc, and this is one sarcomere. But there's many repeating segments of sarcomeres here, which makes this whole thing a myofibril. Now, if y'all remember, we had A bands and I bands. The A band was basically the distance between our thick filament here, right? So this is the A band. And then the I band is the space between two different sarcomeres. So the A band actually gives the dark stripe on the striations, and the I band gives the light stripe on a striation. So if you go back to this slide, and remember we talked about how these cells are striated. They have all these little stripes here. The dark lines in this picture would be the A bands, and the light lines in the striations would be the I bands, which is kind of interesting. But that all goes back to the sarcomeres here within the cell. And those are the functional units of muscle contraction. So if it requires calcium to get the muscle to start contracting, the next question here would be like, well, where does the calcium come from, right? Well, back in skeletal muscle, we said that the calcium came from sarcoplasmic reticulum, which was all this stuff in blue. And that once action potentials came down our T-tubules, it triggered the release of calcium from the sarcoplasmic reticulum. And that's still true in cardiac muscle, because you can see all that here, right? We still have T-tubules. We still have sarcoplasmic reticulum. However, the SR that's here doesn't store as much calcium as you'd find like in a skeletal muscle. And what you also find too, guys, is that some of the calcium that enters the cardiac muscle cell comes from the extracellular fluid. So we're saying that calcium out here, outside of the cell, actually flows in through voltage-gated calcium channels, through a slow-type calcium channel, and then that calcium actually flows into the cell from the extracellular fluid. Okay, so that's another source of calcium in this regard. Now, we also find lots of mitochondria here, and that's going to be important in terms of supplying the ATP to keep these sarcomeres contracting. Because if you all remember, it required a new ATP molecule to get the myosin heads to unbind, and then we can use the chemical energy in ATP to recock the myosin heads into their sort of readied position. And that way, if the actin binding sites were available, they can reach up and grab again and then contract, unbind, recock. And the whole thing repeats, right? And these myosin heads can grab on, causing the whole sarcomere to shorten up. Okay, cool. All right, guys. So some differences with skeletal muscle too, guys, is that the heart does not need your nervous system to contract. So that was unlike skeletal muscle, because if you remember going back to that chapter 9 in AP1, we said that at the neuromuscular junction, we had the release of neurotransmitter onto the motor end plate, where those neurotransmitters bound to receptors, and that could generate an action potential in those muscle cells, right? Now, what we're saying then is that if you need to have neurotransmitter released onto a muscle, that means it requires nervous system stimulation. But that's different in cardiac muscle. Turns out that cardiac muscle cells, some of them, or cardiac cells rather, can generate their own action potentials in the absence of any nervous system stimulation. So what this means then is if all of the nerves to your heart were cut, like every single nerve to your heart were cut, your heart would still beat on its own. But what if you cut the nerves to a skeletal muscle? Would that skeletal muscle still contract on its own? No, exactly. We know this because like if someone has spinal cord injury, they're paralyzed. Like they can't continue to use their legs on their own, like without their nervous system telling their legs what to do. But that's different than their heart though, right? Because if you cut the nerves of the heart, your heart can still beat or contract on its own. And the reason for this is that it has what are called autorhythmic cells. And they account for about 1% of the cells in the heart itself. And these autorhythmic cells are also called pacemaker cells. And the reason being is they can generate their own action potentials spontaneously without the nervous system. Okay, we'll talk about how that works here in a minute. Now remember, all the cardiomyocytes contract as one unit, so it's all or none. So what that means then is as soon as we get action potentials generated in some of these autorhythmic cells, those action potentials are going to spread across the whole heart, and the whole heart is going to respond as one unit, right? Or as it should, right? I mean, it might not under certain disease states, but we're going to talk about how things normally work. So we also find, too, guys, that the action potentials are very long in cardiac muscle cells. Going back to skeletal muscle cells, we said the action potential in those ones was like maybe 2 milliseconds. So 2 milliseconds is like 1/500th of a second. So it's super fast, right? In a cardiac muscle cell, our action potentials are about 250 milliseconds long, which is only 1/4 of a second. So we're saying that these are much longer. And this will make sense in terms of what muscle cells do in the heart, right? In skeletal muscles, we talk about there's muscle twitches. And those twitches can summate and allow you to kind of maybe lift an object, right? So you might have lots of individual twitches that summate and allow you to lift a cake or something, But what we find though in the heart is you wouldn't want the heart to twitch. Because if it only twitched, it would be like just one little flutter, and it wouldn't even pump blood. So what you need then is a nice, long, smooth contraction and relaxation to force that blood out. And what allows for that is this long absolute refractory period, which ultimately relates to the length of the action potential itself. So we're saying it's 250 milliseconds in length. In a skeletal muscle cell, it was only 2 milliseconds. So this is over 100 times longer than a skeletal muscle cell, which is pretty interesting. Okay, so some other similarities, you guys, we find that once those action potentials travel down the sarcolemma, they still have a maximal action potential peak right around 30 millivolts or so. This is due to the sodium channels that are opening as well. That's the depolarization phase of the action potential. Remember, the depolarization is what travels down T-tubules. Again, triggers the release of calcium by the sarcoplasmic reticulum, and that calcium binds to troponin. What does troponin do, though? What's the function of troponin? You guys remember? So troponin is what binds with calcium, right? But do you guys know what troponin does, though? Yeah, good. Yeah, exactly, good. So troponin is what pulls tropomyosin out of the way, right? And so you need to have troponin available and ready and functioning properly in order to get a muscle contraction, right? Very good. So calcium binds troponin, then the filaments slide. Now in this little arrow here, there's a lot of missing steps. This is just assuming that you guys already know about what those steps are, right? But essentially, when calcium binds to troponin pulls in tropomyosin, then the myosin heads can bind with those available active sites on actin on the thin filament, if that sounds familiar. So some other differences, though, here, you guys, is that if you all remember, we said that some of the calcium that's involved with cardiac muscle contraction comes from the extracellular fluid, not just the sarcoplasmic reticulum. And that comes from these slow, voltage-gated calcium channels in the sarcolemma itself, right? In fact, this actually is what contributes to something called the plateau phase of the cardiac action potential, which we'll see here in a minute. In the heart, the action potentials are very different than in skeletal muscle. So you will still have the same phases like depolarization and repolarization, but they can sometimes be due to different ions, and there's different steps in there too. So we'll talk about that. Now, this calcium surge through the slow channels, though, prolongs that depolarization phase such that we actually call this an extra plateau phase. There's a plateau phase of the cardiac action potential where it's sort of long and flat, and this is what lengthens the action potential to 250 milliseconds, right? Instead of only being 2, it's 250 milliseconds long, so pretty long. So some other differences here, you guys, is that if the heart works as a muscular pump, we need these action potentials to be longer such that you can get a nice, long, smooth contraction to really force that blood out. If cardiac muscle cells only just kind of twitched, like skeletal muscle cells do, you're not really pumping blood there. You're just kind of like causing blood to be a little turbulent and move around a bit. So you can get a nice, long, smooth contraction. Now, when I say long, this is still a quarter of a second, right? So 1/4 of a second is still pretty fast. But as far as like cellular activities go, that's actually very long, right? But it's still fast enough to obviously, pump blood and keep up with our body's metabolic needs. So we also have repolarization, you guys, as well here, and that happens when the voltage-gated calcium channels close and the voltage-gated potassium channels open, just like the other action potential, right? So once those voltage-gated potassium channels open, potassium then can leave the cell, and that by removing positive charges, it makes the voltage more negative again, back down towards rest. So what this looks like then is pretty different than what we've seen in the past. So let's go through this together, you guys. So if you guys look here, we have membrane potential or voltage on the y-axis, and then time on the x. So over time, we're seeing a change in voltage. So if you guys notice here, the action potential looks very different. In the past, all you guys saw was kind of like a little line that went up and then down again, and then back down to rest. This one goes up, so we still have the depolarization phase. And just like the skeletal muscle action potential, it's due to the opening of voltage-gated sodium channels. So sodium rushes into the cell, and it depolarizes the inside of the cell by making it more positive up to a point. And then right around this area is when the voltage-gated sodium channels close, and then we have the L type, which is the long or slow voltage-gated calcium channels open. And those voltage-gated calcium channels then add an extra step in here called plateau. Do you guys see this? This long kind of flat part? That was very different than the other action potential we've talked about in the past. This plateau phase is actually due to calcium influx from the extracellular fluid through those L-type calcium channels. So what was the significance of having a plateau in cardiac muscle versus just having a really fast action potential in skeletal muscle? Yeah, longer action potential, which allows for a longer contraction. And why do you want to have a nice long contraction in cardiac muscle? Yeah, there's time to actually pump blood. What if this action potential was only 2 milliseconds, like in skeletal muscle, where it just goes up and then down right away? You're going to get a really brief contraction, right? Can you pump blood with a really brief? No, it's like a really brief contraction, you guys, would be like, I couldn't really open this door, right? I could just do something like this. That's like a brief contraction. It basically did nothing, right? I could just do a brief contraction, and it didn't open the door at all. So what I need then is a nice long contraction to get that door to open, for the blood to leave, and then a nice long relaxation. Just, basically have filling, if that makes sense. Okay. So what we find then too, guys, is that there's a third step in here, which, like the skeletal muscle action potential, it's still due to voltage-gated potassium channels. So in step 3, which is the repolarization phase, voltage-gated potassium channels open, you get a potassium efflux, so potassium leaves the inside of the cell, and by removing positively charged ions, the voltage goes back down towards rest. Okay? So what about during step 1, you guys? What ion's flowing here? Voltage-gated sodium channels are open. Sodium is flowing in. Good. How about in step 2? Voltage-gated calcium channels are open. Calcium is flowing in. Very good. How about in step 3? Voltage-gated potassium channels are open. Potassium is flowing out because you're removing positive charge, which makes the voltage more negative. Okay. So here's the interesting thing too, guys, is that in step 2, all this calcium that's flowing into the cell through the plateau phase, that's the same calcium that's going to go to troponin, bind with troponin. That way troponin can pull on tropomyosin and get the myosin heads to form a cross bridge with actin, right? So that was different than skeletal muscle too, because parts of the action potential are actually contributing to the onset of cardiac muscle contraction, which is pretty interesting. Okay, guys, so In terms of energy requirements for cardiac muscle, we find that there's lots of mitochondria. So the heart is pretty dependent on aerobic respiration. In fact, what we find is that if you have anemia, this can lead to chest pain. So there's a really interesting phenomenon then between kidney failure. Excuse me. There's an interesting phenomenon between kidney failure and chest pain. Because we said that your kidneys release a hormone called erythropoietin if your blood oxygen levels were low. You guys remember this one? And so erythropoietin is what stimulated red bone marrow to make more red blood cells. Now, if you have kidney failure, are you stimulating new red blood cells to be made? No. So then you have anemia, which means you have low oxygen carrying capacity in your blood. But what if your heart is really dependent on oxygen, right? Well, what happens then is that your heart still has to pump blood, but if it's not receiving a sufficient amount of oxygen, that can lead to damage of these cells, which can lead to pain. So you can get chest pain from anemia due to kidney failure, which is an interesting phenomenon there. And the reason being is that your heart is highly dependent on oxygen. So it's dependent on aerobic respiration. Now, it does participate in a little bit of anaerobic respiration. So what's kind of cool too is that your heart can store the lactic acid that enters your bloodstream from skeletal muscle. It can store that lactic acid for later use. We can convert it back into pyruvate, and then that can be converted into acetyl-CoA to feed into Krebs cycle. But there's little anaerobic respiration ability of your heart, which means that you basically need enough oxygen to keep your heart pumping. If you run out of oxygen, your heart will eventually stop. So this is kind of interesting. This is also one of the reasons why hearts can beat outside of the body. You guys ever knew this phenomenon? That if you're doing like open heart surgery or something and the heart's exposed to air, the heart muscle can get a little bit of air from that sort of environment, which means it can still kind of beat just because of air exposure in that regard, right? So it can readily switch to fuel source though. So for instance, it can use lactic acid from skeletal muscles. So let's say if you're exercising and lactic acid is being, released into your bloodstream, your heart can use that lactic acid for energy, which is pretty amazing. It's just like your liver. Your liver stores that lactic acid for later use too. So what we find though, you guys, is that if your cardiac muscle cells become ischemic, or if they have like a lack of blood flow because of a blockage or something, then they undergo anaerobic respiration where they start to build up lactic acid themselves. Now this is a problem because lactic acid is an acid, right? And so if this acid builds up, you get things like calcium dysfunction, right? So you can't regulate calcium concentrations as well. So calcium starts to build up inside of the muscle cells. We also get damage to mitochondria. And by changing the pH of the intracellular fluid, those gap junctions can also start to close up. What were the functions of those gap junctions? Y'all remember? Like, why do we have gap junctions between our cardiac muscle cells? So electrical currents can flow from one cell to the next. Very good. So if those gap junctions close because the cells are acidotic, then what we're saying then is those action potentials won't be able to spread very easily or at all. And what that can lead to then are fatal arrhythmias, and arrhythmia is an abnormal heart rhythm. And that could come from the acidosis itself, which is pretty interesting. All right, guys, so remember we said the heart depolarizes and contracts without the nervous system. Do you remember why that was? Like, how can the heart generate its own action potentials without the nervous system? So we don't need nerves to release neurotransmitter onto the heart. What types of cells can make their own action potentials here? The pacemaker cells, very good. So the pacemaker cells, also called the autorhythmic cells, they account for about 1% of the cells of your heart, and they can basically generate their own rhythm in the absence of any kind of nervous system stimulation. So what we find then is that your heart coordinates as one kind of unit because of the gap junctions and something called this intrinsic cardiac conduction system. So we'll talk about this conducting network, but the conducting system is made of these non-contractile autorhythmic cells. So we're saying here, you guys, is that 1% of the cells in your heart don't contract. Their sole purpose is just to make action potentials. And they can spread those action potentials across the heart. That's their sole purpose in this example. Okay? So they don't contract. Those autorhythmic cells they're not muscle cells. They don't contract. They just generate action potentials and spread those action potentials along. Excuse me. So those pacemaker cells, the reason why they're able to generate action potentials on their own is they have what are called unstable resting membrane potentials. So if we go back to here, you guys, go back to this slide. Do you remember that when we have a flat line down here, we call this rest? This was resting membrane potential, which here is pretty negative. They're saying resting membrane potential is like negative 90 or something, right? Which is typical of muscle. Now, this is what we're talking. This is an action potential in a cardiac muscle cell. But are the cardiac muscle cells the ones that generate action potential spontaneously or on their own? No. Which ones are the ones that generate action potentials on their own? The pacemaker cells or autorhythmic cells. So this is not showing an autorhythmic cell. Okay. So cardiac muscle cells do have a resting membrane potential. Pacemaker cells don't. They're restless. Okay. We'll see an example of this here in a minute. So what we find then, you guys, is that they're continuously depolarizing. The reason why we have continuous depolarization of these cells is they have unstable resting membrane potentials due to something called funny channels. And funny channels are kind of like these leakage channels that allow for sodium and potassium to flow through. But they're more leaky to sodium than potassium. So what we find then is that there's a general tendency of these cells to depolarize up to threshold. And then once they're at action potential threshold, then you'll get an action potential, right? Then it goes back down to what we would call rest, except they don't rest. Because the funny channels will continuously depolarize up towards threshold. We'll get another action potential, right? And where you think that they should be resting, they won't rest, right? They just depolarize up towards threshold again. So that's because of these funny channels here. Now, what's weird though about the pacemaker action potentials is that at threshold, it's actually voltage-gated calcium channels that open. And these voltage-gated calcium channels are what cause the depolarization phase of the pacemaker action potential. Then we get voltage-gated potassium channels that open that allow for repolarization. So again, it's a little different here. Now, the repolarization results from inactivation of calcium channels and the opening of these voltage-gated potassium channels. So the way this works, you guys, is that pacemaker potential is that unstable resting membrane potential due to leakage channels in the membrane of these cells that are leaky for potassium and sodium. So what happens here is that they're more leaky to sodium and less leaky to potassium. And what this allows for then is there's a net influx of sodium at rest, you might say. But because there's a lot of sodium flowing into the cell, it's not staying negative. It's actually depolarizing up towards threshold. It's just sort of leaking in sodium, right? And so if you leak more sodium in, you're going to get to threshold faster. If sodium leaks in more slowly, you'll get to threshold more slowly, right? And so once you reach threshold, then we start this depolarization phase, which is due to the opening of voltage-gated calcium channels. You get an influx of calcium, which causes the rising phase of the action potential of depolarization. Then near the peak of that action potential, voltage-gated potassium channels open, and you get an efflux of potassium, just like other action potentials we've talked about. So let's check this out here, guys. This is what's weird here about this action potential is, do you see any resting membrane potential? Nope. There's no rest here, right? There's just a bunch of lines going up and down. And the reason being, you guys, is that between the action potentials, so if the action potentials are phases 2 and 3 here, right? Between those two, there is no rest. And the reason why there's no rest is because of this pacemaker potential. So pacemaker potential is due to the leakage channels in these cells. So remember, leakage channels are always open. That's why they're called leakage channels. If they're always open, that means they're always leaking. And so imagine then that these channels are leaky to both sodium and potassium. Now, sodium wants to flow into the cell. Potassium wants to flow out. So if you had equal amounts of sodium and potassium flowing in and out, then there would be no net change in voltage, right? Because you're removing one and adding one, right? So, but what we find, though, is that these pacemaker potentials are more leaky to sodium and less leaky to potassium. So what we find then is that there's actually a net influx of sodium versus potassium. So you're bringing in more positively charged sodium than potassium, okay? And so what this causes then is a net depolarization up towards action potential threshold. And at this dotted line right here, at threshold, this is where the voltage-gated calcium channels open. You get a calcium influx, which causes this phase. and then we get the voltage-gated potassium channels that open, which then cause repolarization back down towards what we should call rest. But there is no rest, right? Because as soon as you go down to the bottom here, what happens next? Once those voltage-gated potassium channels close, what takes over? Pacemaker potential. And you slowly start to depolarize up towards threshold, and you get another action potential here, again, towards threshold. Now here's the interesting thing, you guys, is that pacemaker potential, the slope of that line is modifiable based on your sympathetic and parasympathetic nervous systems. So what your sympathetic nervous system would do then is make these pacemaker potentials more leaky, like make these funny channels more leaky, rather. So if you made the funny channels even more leaky to sodium, would you get to threshold faster or slower? What do you guys think? Yeah, faster, exactly. And if you get to threshold faster, what happens to the frequency of action potentials that are generated? You increase frequency. Very good. So if you increase frequency of action potentials, what's going to happen to your heart rate? It's going to go up. So that's how your sympathetic nervous system increases heart rate, is your sympathetic nervous system makes these pacemaker potentials have a steeper slope such that you get to threshold faster, which means you can fit more action potentials in a smaller amount of time, which is an increase in frequency. And by increasing the frequency of action potentials, you increase your heart rate. So what do you guys think your parasympathetic nervous system does if it slows down your heart rate? Yeah, very good. So your parasympathetic nervous system makes the pacemaker potentials less leaky to sodium, such that the slope of these pacemaker potentials is actually lower, and it takes longer to get to threshold. If it takes you longer to get to threshold, what happens to action potential frequency? It goes down. And if your action potential frequency goes down, what about your heart rate? It also decreases. Very good. Make sense, you guys? Okay, so your book does go into that mechanism. We won't go into it in too much detail here other than what I just mentioned, but kind of keep that in mind, though. And that's how you can change your heart rate, is you're basically changing your pacemaker potentials. Now, the reason why we go over this, you're thinking, well, why do we even care about all these voltage-gated channels, that kind of stuff? The reason why we care about this stuff, you guys, is that a lot of the drugs we use to target the heart for like arrhythmias or blood pressure, they're going to act on these types of channels. So you might give someone a calcium channel blocker to prevent, hypertension, because it's going to prevent that plateau phase from being too long, such that your contractions are a little weaker, right? And so there's also lots of diseases where people maybe like inherit like a defection or like a defective potassium channel or defective sodium channel, that kind of stuff, and that can lead to arrhythmias, which we'll also talk about later, like on Thursday. Okay. So what we're going to talk about next, you guys, is the sequence of excitation across the heart. So remember we said that all of these action potentials that are generated spontaneously occur within the autorhythmic cells of your heart. Now these autorhythmic cells are located within regions where they're kind of like in little clumps, right? So what you find then is that the SA node here, the sinoatrial node, is a region where you find a big aggregation of autorhythmic pacemaker cells, okay? Now the SA node is located right near the juncture of your superior vena cava and your right atrium. So it's up top on your heart. And we think about the SA node, you guys, as being the one that starts the pace of the whole heart. And the reason being is that the SA node generates action potentials the fastest. So it generates action potentials at 100 times per minute, okay? In the absence of any nerve stimulation, SA node is 100 times per minute, okay? Then what happens is that from the SA node, action potentials are spread across the atria through those muscle cells, which we call the internodal pathways, where they go back to another node called the AV node. And the AV node is the atrioventricular node, and it's located right at the superior portion of the interventricular septum and on the inferior portion of the interatrial septum of your heart. So pretty much right at the juncture of your atria and ventricles, okay? Now the AV node is also another aggregation of autorhythmic cells, except for these ones generate action potentials at about 50 or 60 times a minute. So it's not as fast, okay? Then from the AV node, it reaches something called the AV bundle of His, And this is where action potentials are delayed by about 0.1 seconds, so 100 milliseconds. And then that AV bundle then branches into the right and left bundle branches, where then electrical currents are spread across the ventricles along the apex through what's called the subendocardial conducting pathway, aka Purkinje fibers. So this sequence is important, guys. It always goes from the top down, right? So the SA node is on top of your heart, the Purkinje fibers are down by the ventricles. So it kind of works its way down here. Now what happens, guys, is that the electrical impulses that are generated can spread across your heart in about 220 milliseconds. So that's less than 1/4 of 1 second. So that's still pretty fast. But this happens to be slow enough such that your heart still has time to pump blood effectively. Okay. All right. So let's start with the SA node first too, guys. So the SA node is the pacemaker of your heart. And the reason why we call this the pacemaker, like the pacemaker of your heart, is that it's the fastest. So by being the fastest, it sets the pace for everything else, right? So its inherent rate is 100 times per minute. So let's say if somehow like all the nerves to your heart were cut and you didn't have adrenal glands to stimulate the heart, then basically the SA node would generate action potentials at 100 times per minute. Is that like a normal resting heart rate? Should you find like someone's normal resting heart rate at 100? No, it's pretty dang fast, right? So what we find then is that normal resting heart rate is slower. You know, you say like 75, as long as it's somewhere between 100 and 60, then we consider that normal actually. So what this means, you guys, is that your normal resting heart rate is actually lower than the intrinsic rate of your SA node because of the fact that you have parasympathetic nerve stimulation to your heart at rest. And your parasympathetic nervous system slows down your heart rate, right? But if you cut the vagus nerves to your heart rate would speed up and maybe even stay right around 100 because of the intrinsic rate of the SA node, right? Okay, so what happens here next is that the impulses that are generated by the SA node, they spread across the atria. So once they generate, they spread across the atria, they go across those atria, and they kind of work their way around towards the AV node. Now, in between the SA node and the AV node, we have what are called the internodal pathways. But the internodal pathway is made of cardiac muscle tissue. So remember, the SA node is what's going to generate pacemaker action potentials. But the pacemaker action potential, once it gets to a cardiac muscle cell, it will generate a cardiac action potential. And those will spread across the atria until they get to the AV node, where you're back to another pacemaker action potential. Okay? So the AV node is just inferior to the interatrial septum. It delays action potentials by 0.1 seconds or 100 milliseconds. So why might you want to delay here, guys? If action potentials start in the atria and those spread across the atria, why would you want to hold those action potentials briefly before they move on to the ventricles? Say it again. Yeah, allows time for the atria to contract fully before the ventricles begin to contract. Because what do you guys think would happen if the atria started to contract when the ventricles began to contract? Not good things, right? Not good things, because now they're opposing each other. So what you want then is a nice atrial contraction, right? Forcing blood in the ventricles, then the ventricles contract. And so by having a delay, it allows for that time. Okay, so that way they don't like kind of step on each other's toes, so to speak. So what we find, you guys, is that the reason why there's a delay here is that the fibers have a smaller diameter. So it's kind of like an anatomical bottleneck. So by having a bottleneck, things kind of back up right there, that place where it's more narrow. So there's less current at those smaller fibers. So it turns out that the AV node generates action potentials right around 50 times a minute. So could you live without an SA node if you still have an AV node? Absolutely, right? But what's your resting heart rate going to be? Probably something less, right? Because you're generating action potentials even more slowly than you normally would. So if the SA node generates an intrinsic rate of 100 and the AV node generates an intrinsic rate of 50, if you lost an SA node because of like ischemia, like a heart attack or something, and you still had your AV node intact, your AV node would generate action potentials at 50 times per minute. Is that normal? Like, is that a normal resting heart rate? No. Not unless you're like a trained athlete. In fact, if it's less than 60, we call that bradycardia. right? So it's a slower heart rate. So the AV bundle of His, or AV bundle, is also right beyond the AV node, and it's basically going to take those action potentials down the interventricular septum before it branches into the right and left bundle branches. Now the right and left bundle branches go down the interventricular septum, and they basically carry those impulses to the right and left ventricles respectively, right? So each bundle branch goes to the right or left side of the heart towards the ventricles. Now, once those bundle branches reach the apex, they start working their way up along the lateral border of the heart, and then all along the way after the apex, those bundle branches start branching out into what's called the subendocardial conducting pathway, aka Purkinje fibers. Purkinje fibers are the end of the road as far as the conducting pathway goes. So after the Purkinje fibers, they basically stimulate muscle cells to begin to contract, and this starts at the apex. Now, even the Purkinje fibers can generate action potentials at 30 times a minute, right? So you could potentially also live without an AV node. So if you lost your SA node and lost your AV node, your Purkinje fibers could still generate action potentials at 30 times a minute. But are you going to be conscious with a resting heart rate of 30? Probably not. Probably not. It could be enough to kind of keep your tissues alive, but you probably won't be able to keep your brain awake. You know what I mean? So you're probably going to be unconscious at this point. So it's kind of interesting. Now, ventricular contraction is what immediately follows the Purkinje fibers sort of stimulation here. So if you guys look at this pathway here, we're gonna see this on the heart. So it all starts with the SA node. So remember, if you want to find the SA node, it's right near the juncture of your superior vena cava, near the right atrium, and the SA node generates action potentials first. So why do we call the SA node like the pacemaker of your heart? It's the fastest. Very good. So it generates action potentials the fastest of any other pacemaker cells of your heart, right? Now, once those action potentials are generated, they spread across the atria through the atrial muscle. So, for instance, you guys, I got a question for y'all. So, what type of action potential are the SA node cells making? Is it a cardiac action potential or a pacemaker action potential? Pacemaker, and that's very different than a cardiac action potential, right? So those pacemaker action potentials then stimulate the muscle cells of the atria to start to make cardiac action potentials. So these dotted lines here, guys, this is showing the internodal pathways. And those, excuse me, those internodal pathways are carrying action potentials through the cardiac muscle cells, but they're making cardiac action potentials, like the ones with plateaus, because these muscle cells are actually contracting as a result, okay? And then once it gets to the AV node, what type of action potential is going to occur here? It's a node, right? So AV node is pacemaker action potential. Because what's the pace of the AV node? Do y'all remember? 50, yeah. So 50 times a minute. So again, they're autorhythmic cells at about 50 times a minute. Then it goes to the AV bundle, where it branches into the right bundle branch and the left bundle branch, right? So the right bundle branch goes down towards the apex, as does the left. But you guys see that through the interventricular septum, there's not a whole lot of little fibers coming off? That's significant because we don't want the ventricles to contract superiorly first. We want to wait for contraction to begin at the apex and then work its way up towards the lateral aspects of the heart, going superiorly. Because if the apex contracts first, that means it's going to squeeze that blood up and out. What if actually, I'm just kidding, up and out this way. That's the atrium. It should go through the aorta. So what if the superior part of your ventricles contracted first, and it contracted from the top down? Can't hear you, I'm sorry. Yeah, the blood wouldn't pump out. You're right, exactly. Like the blood would just kind of get forced towards the bottom of your heart. So it makes sense then that you want to have contractions from the apex working up. That way you can squeeze that blood up and out of the heart, right? And so this makes sense in terms of the anatomy of this conducting pathway, because you don't really see a lot of Purkinje fibers until you get to the apex. So these are the Purkinje fibers, aka the subendocardial conducting plexus. And this is what actually can excite the cardiac muscle cells of your ventricles to begin to contract, okay? So the atria contract first due to stimulation of the SA node. Then those action potentials reach the AV node, where they're held up there for how long? So how long are the action potentials delayed at the AV node? 0.1 seconds. Very good. So 100 milliseconds. And what's the significance of that? Why hold those action potentials up there? Allows time for the atria to contract. The atria then force that blood in the ventricles. Then the action potentials reach the ventricles, where the ventricles contract, starting at the apex, and then the ventricles can squeeze that blood up and out. Now, as the ventricles contract, though, you guys, what happens here is the AV valves begin to close. So when those AV valves close, they create a heart sound, which is the first heart sound, which is lub, S1. That's due to the closure of AV valves. But remember, if I said that whatever's going on one side is also what's happening on the other. So even though I showed the AV valve closing on the left, the right AV valve is also closing. Because you guys see the anatomy is pretty much identical here, where the bundle branches go to both sides. And so that means the action potentials are going to reach both ventricles approximately at the same time, which means that both ventricles are going to contract at around the same time, okay? Which means both valves close at the same time. So that's important to note. Okay, guys, so what this slide shows then is the sequence of action potentials across the heart over time. So if you guys notice, we talked about how action potentials begin at the SA node. They go through the internodal pathways and then stimulate the atrial muscle along the way. Then they reach the AV node. The AV node then sends action potentials along the AV bundle branches, Purkinje fibers, and then eventually ventricular muscle here, which you guys can see here. So it makes sense how they're all kind of delayed, right? Because you have to go from one area to another, to another, to another. Now, you shouldn't find that all these action potentials overlap because there's a delay between the sequence of excitation, right? So what was the whole significance of having a delay between the sequence? So why don't we want the atrial muscle contracting at the same time as ventricular muscle? Do you guys notice that there's a difference there? The action potential here of the atria is very different in time than the ventricular action potential. So why does that make sense in terms of blood flow? So they don't work against each other, right? So the atria contract first to force that remaining 20% of blood in the ventricles. Then once that blood's there, the ventricles will contract, okay? So here's the interesting thing too, you guys. Let's go back here. Why don't action potentials just go from the atria straight into ventricles this way? I mentioned this last week. Maybe you guys don't remember that. I mean, we talked about it a lot last week, right? But there's a reason why action potentials don't just go down this way, straight into the ventricles. And it has something to do with this area right here. Do you guys remember we talked about the cardiac skeleton? And the cardiac skeleton was like the framework by which muscle cells attach to. But I also mentioned that the cardiac skeleton is what holds the valves in place, right? And all those valves, like the AV valves and the semilunar valves, they're all in the same plane. So if you cut the heart the right way, you see all those valves. You guys remember that picture we saw all the valves, and there was all that kind of whitish tissue around the valves? That white tissue was the cardiac skeleton. But the cardiac skeleton is also an insulator. Insulators have low electrical conductance, and they have high electrical resistance. So what happens, you guys, is that action potentials can't go this way into the ventricles, along the sides because there's an insulator right here, which is the cardiac skeleton. So they can only go along this electrical conducting pathway, like under normal circumstances, right? There are some conditions where that's not going to be the case, but under normal circumstances, because of the cardiac skeleton, you have a nice little insulator right here, which prevents action potentials from spreading in the atria straight into ventricles. If they did, would your action potentials look like this? If the action potentials could go from the atria straight into ventricles, would you have this separation of action? No, exactly. They would overlap, right? And that would be terrible, because then what would happen then is your ventricles would begin to contract as your atria are contracting. So that's why we have to have this anatomy to prevent that sort of overlap of contraction. It can happen under certain disease states, but under normal circumstances, no. Cool. All right, guys, so I also mentioned how your parasympathetic nervous system and your sympathetic nervous system attach to the heart. So we talked about this with respect to regulating heart rate. So you guys notice here that in purple is the vagus nerve, which is cranial nerve number 10, and you have one on each side, right? So it comes off the brainstem, right around the medulla oblongata. Vagus nerve goes down your neck into your thoracic cavity, where it has little branches that can go to the heart. Now, we already talked about the vagus nerve in terms of going to your digestive organs, because that's the one that can mediate digestion, and it kind of begins the cephalic phase of digestion. But we also talked about how the vagus nerve can go to your heart as well. Now, if you guys notice here, these purple branches of the vagus nerve only go to the nodes. So if they only go to the nodes, that means your vagus nerve can only change your heart rate and not the force of contraction, okay? Because they're not going to the muscle, they're just going to the nodes. So your vagus nerve can only slow your heart rate down. So what would your heart rate be if you cut the vagus nerves? Fast. But what would it be though? At least 100, right? So why would it be at least 100 if you cut the vagus nerves? Because the vagus nerves normally slow down. Good. But why 100? That's what the SI node generates it at, right? Why do I say at least 100 if you only cut the vagus nerves? Because what else would you have available? The pacemaker cells are still there, and so they're going to generate at 100, right? But that's why it's at the minimum 100. But it could be faster, because if you only cut the vagus nerves, you also have sympathetic nerves available here too. So you guys see here that the sympathetic nerves that come from the thoracic spinal nerves also go to the heart. Except for the sympathetic nerves not only go to the pacemakers, but they can also go to cardiac muscle tissue. And so this cardiac muscle tissue then can also be modified by your sympathetic nervous system to cause your heart muscle to contract more strongly as well. Okay? So your sympathetic nervous system can speed up your heart rate and alter the force of cardiac muscle contraction. So you might want your heart muscle to contract more strongly to be able to force more blood out. Okay? Cool. Awesome. Does that make sense to you guys? Okay. So which one only changes heart rate and not force of contraction? Parasympathetic. Good. Which one can change both? Sympathetic. Good. Now, when we talk about changing the force of muscle contraction, that's what's called an inotropic agent. So your sympathetic nerves would be a positive inotropic agent because they're increasing the force of heart muscle contraction. Okay? All right, cool, guys. So now we're going to move on to talk about EKGs or ECGs. Now, an ECG is an electrocardiogram. The reason why we say ECG or EKG is that cardio with a K is the German form, but we can still call it that too. You can still say electrocardiogram with a K. But to write it out in English, we spell cardio with C, not a K. So you can say ECG or EKG. I prefer EKG just because it's faster and easier for me to say, but either is fine, right? So what an electrocardiogram measures, and we'll actually be doing this in lab today, is the changes in electromagnetic fields of your body in response to all of the action potentials that are occurring in your heart, okay? So the reason why you can measure this, excuse me, is that when action potentials are spreading through the heart, you're moving charged ions, right? And those ions have a charge, like positive charges. And when you move these positive charges around, that generates electromagnetic fields. So if you move a charged particle or something, it generates electromagnetism. Okay? And so you can measure this. It's not a lot, but it's enough to be measurable. So what we're saying then, you guys, is but when the action potential is spread across your heart, and all that sodium's flowing in, all that calcium's flowing in, and potassium's flowing out, by moving these charged particles, you're generating electromagnetic fields around the heart. Okay? Now you can measure those fields across your skin using a recording electrode. And so what an EKG does is it measures or compares the differences in those fields across different points. So with an EKG, you have what's called Einthoven's triangle, where you're basically measuring or comparing the points across the limbs. So today in lab, we'll be attaching electrodes to our limbs, and the EKG then will measure and reference across those points. So it's going to compare the left upper appendage to the right upper appendage, or the left upper appendage to the left lower appendage. And it's referencing those points because those fields are asymmetric in terms of location of your body. then it's going to have difference in reference points here. So an EKG is not measuring individual action potentials. It's measuring all of the electromagnetic fields that result from heart muscle contraction. But what if other muscles were contracting in your body? Could you isolate just the effect of the heart? No. So what does that mean if you're trying to get a good read on someone's EKG? They got to be still. They can't be moving very much, right? Because if they're moving a whole lot, and let's say if they're using the muscles of their arm, well, guess what? Those muscles generate electromagnetic fields too. It's not just your heart. So if you're using your arm muscles while someone's trying to get an EKG, you're not going to read it very well. So what that means then is today in lab, when we read the EKGs or try to take some, you got to stay very still, right? Don't even breathe very heavily. Just kind of stay still, and we'll get a nice kind of clean read on that too. So it's pretty awesome. All right, guys. So with the EKG, we're measuring the electrical events of the heart, and they occur in three kind of key phases. We have our P wave, QRS complex or wave, and our T wave. Okay. So what this looks like is this. So the P wave on an EKG is this upward deflection. But what's important to note, you guys, is that the upward deflection doesn't mean like depolarization or repolarization. So remember when we looked at the action potential? If the curve deflects upward, that means depolarization. And if it deflects downward, that means repolarization. That's not the case with EKGs. And that's because of just how they're measured. So these deflections just mean a change in, right? So the change could either be depolarization or repolarization, but the deflection just represents a change, not the direction of that change, if that makes sense. So what we find then is that with the P wave, this is what correlates with atrial depolarization. So remember, depolarization is when action potentials are beginning. So that P wave is going to be when the action potentials begin to spread across the atria. Okay? So what should happen next after that? If the action potentials spread across the atria, what should be the next event? What's that? They should get to the AV node, and then what? Then they go down the bundle of his to the bundle branches along the ventricles, and then what? The ventricles contract. Good, exactly. So as those action potentials spread across the ventricles, that's what causes the QRS wave here. You guys see that? QRS. So QRS complex here correlates with action potentials spreading across the ventricles. But do you guys notice there's a delay between the P wave and the Q wave? It ends up being about 100 milliseconds. Why was that? What caused the delay between the atria and the ventricle contraction? 100 millisecond delay. The AV node. It's a bottleneck. So what about the AV node made it a bottleneck? Do you guys remember? That's good. That's good. Yeah, you're right. So it allows for time for the atria to contract before the ventricles start contracting. That makes sense. But because of the reason why this delay exists, you guys, between the P wave and the Q wave, called the PQ interval, what we're saying there is that that's because the AV node is an anatomical bottleneck. The fibers are very narrow there, so they don't conduct as much current or as quickly. That's what allows for that 100 millisecond delay. And you can see that 100 millisecond delay on the EKG, like a normal EKG, right? Cool. So there's our QRS wave. So why do you guys think the QRS wave is so much bigger than the P wave? If the P wave is the atria and the QRS wave or complex is the ventricles. This one's way bigger, right? Like there's a bigger signature there. How does that make sense in terms of the anatomy of the heart? There's more force, there's just more muscle, right? There's more ventricular muscle than atrial muscle. Like atrial muscle was super thin, there wasn't much of it there, right? So the QRS complex basically represents the ventricular depolarization here. Now in the QRS complex, you would also find atrial repolarization, but you're not going to see it because it's kind of masked by the effect of the ventricles. Like they have a louder signature here, okay? And then we have some delay here, and then finally we have ventricular repolarization. But repolarization means the action potentials are going away, right? right? So what mechanical event should correlate with ventricular repolarization? What do you guys think? If the action potentials are leaving the ventricles. Are the ventricles still contracting? No. Good. So what's happening then at this point? Like ventricular repolarization. The ventricles are relaxing. Very good. So here's the key point, you guys, and this is going to be really important before we meet again on Thursday, is I need you guys to review this, and we'll have a group activity on this. It are the correlation between the electrical events of the heart and the mechanical events of the heart, right? And that's in something called a Weiger diagram, which I'll show you guys here in a minute. But I need you guys to review that Weiger diagram before we meet on Thursday, because we'll have a group activity on that. And remember that oxygen hemoglobin dissociation curve we talked about? It's kind of like that. Like, dang it. There's all these curves. Like, okay, but if you can understand that diagram, you know the heart, right? Because it's showing the electrical events with the mechanical events, with the heart sounds, everything that happens in one heartbeat. So it's pretty important to know too. So what does the P wave correlate with again? Atrial depolarization. How about the QRS wave? Action potential spread to the ventricles, but specifically ventricular depolarization, which is the onset of contraction. Good. And then how about ventricular repolarization for the T wave? What does that mean? The ventricles are beginning to relax because the action potentials are repolarizing, right? They're becoming more negative, so they're going away. Awesome. Good job, you guys. So if we can look at that here, we're seeing that in yellow represents, or orange rather, represents, what is that yellow-orange? I don't know. So represents depolarization. And then red here represents repolarization, right? And so what we're saying to you guys is that the P wave is when action potentials begin to spread across the atria. You guys can see that here, right? And then those action potentials have a slight delay at the AV node, which is what we see here with the PQ interval, which is about 100 milliseconds or so. Then, after those action potentials go down the bundle of His and down the bundle branches, and you start to get ventricular depolarization, we get our QRS complex. And then once the action potentials spread across all the ventricles, we get this little delay here between the S and the T wave. And then as the ventricles begin to repolarize here in red, you can see our T wave begins. And once the ventricles are totally repolarized, now we're sort of in a resting period between the next heartbeat. Okay? So all this happens pretty quickly though, you guys. There's no time scale here, but it's pretty rapid. Okay. So looking at some abnormal and normal EKG tracings here, we can see here is like a normal sinus rhythm. And the reason why we can tell it's normal is that the interval between all of these QRS peaks is about the same. Okay. So there's a really fast way to determine heart rate by looking at an EKG here, you guys. And the reason being is that all of these little squares represent time. And the time is pretty standard, okay? So the way you can calculate heart rate by looking at an EKG here is each of these big squares here represents, it's 0.2 seconds. So if you guys look here, we have 1, 2, 3 big squares. So you're saying that there's basically a heartbeat every 0.6 seconds or so. But there's an even easier way to figure this out. If you count the number of big squares between each heartbeat, and it happens to be 3, and divide that by 100. I'm sorry, no, take 300 and divide the number of big squares into that. So what's that gonna? I gave the answer away. Dang it. that was my problem. I was doing the math as I was describing how to do it. So it's 300 divided by 3, which is 100, right? So 300 divided by 3 big squares is 100 beats per minute. So that is technically normal sinus rhythm. Why is that? It's not tachycardia. So why is it considered normal if it's at 100? Close. It's the SA node, right? Because the SA node normally generates it at 100 times per minute. So you can have a resting heart rate of 100, and it's still technically normal. I mean, it's the higher end of normal, right? So you're probably concerned at that point. But you still wouldn't say tachycardia, though. It has to be over 100, okay? So that makes it pretty straightforward to just quickly identify, right? So take each of these peaks here, count the number of big squares between each one, and divide that number into 300, and you'll basically get your heart rate. But that only works if it's a normal rhythm, right? So if there's a difference between the distance between these peaks, you know, and it's not a normal rhythm, you couldn't do that because you're not going to get an accurate value there. Okay, because sometimes you might find only 2 squares between them, or you might find 6 squares between them, right? So then you can't just do one and just know, oh yeah, for sure that their heart rate's that. Okay, okay. So what we have over here, you guys, is something called a junctional rhythm. That's where the SA node's non-functional, and instead, what's happening then is you don't get the normal P waves as you would normally see because the action potentials are beginning at the AV node, not the SA node. And because they're beginning at the AV node, that P wave is kind of hidden within the QRS wave. Okay? So the atria are still contracting. It's just that they're contracting, you know, too close to when the ventricles are contracting, so that's kind of masked within that QRS wave. So you get to see the QRS and then our T wave here. That's a junctional rhythm where the SA node is non-functional. And then with a second degree heart block here, you guys, what we're seeing is that there are multiple non-conducted P waves. Okay, so what this means then is, if you look up on this graph, you guys, we have P, QRS, T, right? So P, QRS, T. But then here's a P wave, and nothing happens. You guys see that? And then we get a P, QRS, T, P, P, QRS, T. What should have happened after that P wave here? A QRS wave. That's why it's considered heart block, because there was a non-conducted P wave. So there was an atrial contraction because the action potential spread across the heart, but it didn't stimulate the ventricles to contract. That's why we consider it a heart block. Now, don't worry about second degree or whatever, because we'll go through the types of degrees of heart block in pathophysiology, but just kind of keep that in mind. And this is showing an example of VFib, ventricular fibrillation, where basically the ventricles are just sort of, just kind of like contracting really rapidly. And you can see that just represented here just as this crazy sort of lines here. So kind of cool. All right, guys, so we'll stop there for today and then